Howdy, I'm Rob Orr. I've been teaching at OES since 2002 and teaching science since 1980. And I wanted to give you a quick introduction or background on the SRP program, the Science Research Project program. That's the hallmark of what we do. Right now I'm going to walk briefly around the project room, which is one of the facilities that we have, which has a lot of support for a lot of wide range of the different projects. We have environmental chambers, we've got a woodlands right back here. On campus we have a wetlands area, we have 2 streams running through. We have 3 classrooms, a drop zone, an instrument lab, and a full dark room up top. So we've got a range of different facilities, but what's most important is we have a really highly talented support staff, not counting me, but we've got other science teachers who are very talented, and we tend to really all rally around everybody's science project. Science is messy. The way that we get students to do it is every student, 140 in the fall and 70 in the spring, who are taking full year-long science classes have to develop and propose and get approval for a science research project that they carry out themselves independently. And that involves a lot of troubleshooting and a lot of initiative and a lot of bad data frequently. And that's one of the issues that you run up against, but that's what science is. Science is rarely, coming down from a mountaintop with a great answer. So we have the facilities here, including supervision passively through the window to the science office, where I'll pound on the door and say, put your goggles on. But generally we have good support after school. This year we're trying a whole lot more at-home science being done, and that can also be done in the field and other places.
Hi, my name's Peter Kimball. I'm a science teacher here at OES, and one of the things that OES prides itself in is training our students to do really good science. And that has to start with observations and good questions. So I'm going to show you a little demo here. I'm going to have you do just that as you watch. Make some observations and ask some questions. Okay, here we go. So you might notice here that we have a pretty large Erlenmeyer flask, and we have this orange substance at the bottom. I'm going to take this clear liquid from this also massive beaker and just pour it right in there. And immediately we notice that the color goes from that orangey color to this deep, beautiful indigo. We're not done. I'm going to get this stir bar going here. And I'm just going to top us off with a different clear liquid. Here we go. Keep asking questions and making observations as we're going here. Still retaining that indigo color. You saw flecks of green, you might have seen some yellow, and now we're back to that orange color. If I add a little bit more here, we'll go to that deep red. And then, as a special treat, let's head right on back to indigo. So, questions you might be asking yourself. How does this work? How do you take clear liquids and add them to a substance and for some reason it changes color? Is this a physical change? Is it a chemical change? What would happen if we messed with the temperature? What if I turned off the stirring rod? All of those are good questions. But the one that I'm going to leave you with here is this magic or is it chemistry? And my hint, not so subtle, it's not magic.
I'm Taposhi Biswas. I teach physics at OES, and I'm also the Winningston Chair for Science and Engineering. Today I'm going to show you a gas discharge tube demo. Basically, we're passing electricity through these little vials of gas, and they're going to produce pretty colors that are going to be a mixture of a bunch of different colors. And then we're just going to use this little diffraction grating. Different colors of light travel at different speeds through this grating, so it'll basically split the colors coming out of these gas discharge tubes, and that will show us something about the structure of the gas that is inside the tube. We're actually going to put the grating right in front of the tube here, and you'll be able to see the light coming through, and there's sort of a salmon-y color that's coming off of the tube, but when you look through the grating itself, you should be able to see a couple of different images of that tube so that it looks like they're different colors, and you can see all the different colors that are being put out by that tube. So then the fluorescents are actually sort of the same setup as these little gas discharge tubes. They're obviously a lot brighter, but they have essentially mercury vapor in there as opposed to, you know, the water and helium that are in these tubes, and so it produces a different spectrum. So you can still see these multiple images of the fluorescence, but it's different colors than you would see in the gas discharge tubes.
So today we're going to look at 3 demonstrations, and it's your job to figure out what these have in common. Now it's possible that you already know what they have in common, and that's fine, but just keep it to yourself for now. But the next part of the questions we're going to explore will ask you to think a little deeper. And that has to do with, I'm going to ask them what would happen if questions, and you're going to have to predict and then explain why. So let's start. Okay. So that's cool, okay? What would happen if we used smaller rings? Would it make it easier or harder? What would happen if we used this much sand instead of this much sand? That's for you to figure out. So we have 30 pennies. We'll see what happens. Now, what would happen if I used 2? Would that make it harder or easier? What would happen if I used pre-1983 pennies that are pure copper and much heavier than post-1983 pennies that are much lighter? What would happen? How do you know? Explain why. So for this one, we have this huge thing of water. It looks fancier, but it's just water. In a glass, I promise, beaker. And we're gonna try something. So I'm just gonna step around here. So now we're gonna, this is getting to the edge. Woo! What's gonna happen? Magic, or chemistry, or science. What would happen if I pour water out? And we try it again. Would you do it? Why? Why not? Tell me.
Hey guys, my name is Bettina Gregg. I teach honors biology and genetics. I used to be on your side of the desk. I'm an alum of the class of 92, but now I'm enjoying being on this side and being able to teach you everything I know about biology, about biotech, about genetics, and all these fun topics that I love to talk about. Today we're going to look at the sleep lab, which is one of our student favorites, because you get to take a look at what your own genetic makeup is when it comes to the period gene, meaning one that suggests whether you are morning people or night owls. And it's always a really fun thing to look at your own genes. We have our samples ready right here in our thermocycler, which means in the last several hours, this machine has helped us by making millions of copies of our DNA sample, so we're ready to take it out and use it. So I'm gonna take my sample and I'm going to grab a pipette tip. Remember to keep it clean, keep it sealed. Take my 10 microliter sample, which I know is hard for you to see. Close the tube, and I'm going to take it and load it into my electrophoresis gel. All right. I'm going to close up my box, and now as I run the electricity through this, it's going to separate out my DNA sample. I'm going to turn the machine on, and in about 45 minutes we should find out whether we have morning people in this group or night owls or a combination of both. Well, thank you for joining us on this short tour through our science department. There are so many more things we could be exploring, but our time is limited. So much more fun to be had, and I hope you enjoyed this little glimpse into all the fun we have around here.